I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
For the November Cranberry Virtual Brown Bag, we take a couple of months off for harvest, and then we group back together post-harvest to hear all through the growing season. We've heard a lot from our Wisconsin specialists, but we want to make sure that we're plugged into the rest of the nation and the rest of the cranberry growing world as well. So today we're going to hear from researchers out in British Columbia and in Massachusetts and in New Jersey to make sure that everything that is going on, we're getting to learn about and take advantage of and stay connected with. So we're looking forward. I want everyone to feel comfortable asking questions. As far as asking questions, you can do either the chat, which I'm monitoring and will ask on your behalf. You can also unmute yourself and ask a question directly. And if you want to ask a question anonymously, you can send a chat message that says just to Allison, and I will ask that question anonymously. So lots of ways to interact. I also want to mention that we need to remind there have been lots of tax changes in this year that will impact the next taxes you file. So I was just asked to say, if you have not yet talked to your tax specialist, you're a little bit behind the 8 ball. Please talk to your tax specialist. After those little housekeeping bits, I want to thank Tom of the Growers Association for helping host these. So Tom, if you want to say good morning.
Well, technically, I'll say good afternoon, and welcome to our continuing virtual brown bags. And thanks to Allison and Alex for putting these together. It's a great opportunity for growers to get together and take a little bit of time to learn on topics. So we try to hold these that have relevant topics, and at times that growers can join. I know a number of you are probably considering getting out into the woods or looking at it this weekend, and we wish you well with that. But I just want to say welcome, and we're going to continue to do these types of programs. We did have one thing to make folks aware of. We did start the process for developing the research program for the upcoming year with the research roundtable that was held on Wednesday, and we had very good participation, and I think we had a good session. That leads to the Wisconsin Cranberry Board annual meeting, which will be held, somebody correct me, but I believe it is the 29th of November. And but everybody should have gotten a notice on that, and there is a virtual option, so we welcome you to join or come down to the research station, join us at that. But just welcome everybody, and if you're going hunting, have a safe hunt, and hope you and your family have a great Thanksgiving.
Thank you. Thanks to feedback from the education committee, from growers, and from researchers at Wisconsin, I kind of took a little straw poll of who we really wanted to hear from at these brown bags. And a reminder that if you ever have someone you want to hear from, let me know, and I'll do my best to book them in. But we had questions about pollination deficit, which is an area of expertise for Sandra Gillespie at the University of Fraser Valley. We wanted to hear Jeff Neihart is new at the genetics program in New Jersey, and so we wanted to hear what he's looking forward toward doing. And then Giverson Mupambi has been doing a lot of work in fruit color and quality. And fruit color and quality also came up a lot at our research roundtable on Wednesday. So those are important and interesting topics. So I'm hoping everyone is looking forward to hearing them. Giverson, if you want to share your screen.
Okay.
Fantastic. A little bit on color and quality.
All right. Good afternoon, everyone. Thank you for the opportunity to come and present some of the research that I've been doing for the past 2 years. Since I'm new to many of you, I'm going to start by giving a brief overview of some of the research projects that I've been doing. I started my position in 2019, and I do applied research focused on plant physiology. For those of you not familiar with the Zimbabwean accent, I try to put as much information on the slides as possible, so if you can't follow me. All right. So one of the things that I'm actually working on is the evaluation of new varieties. Here in Massachusetts, we do not have a breeding program like you guys in Wisconsin or in New Jersey. So I've been working on the evaluation of new varieties and trying to give some of our growers performance data that is based on our conditions. Another project that I'm currently working on is agrivoltaics. So agrivoltaics is the co-location of agriculture and renewable energy production. We started this project in 2019. We have those plywood solar panels that you can see on the left. And currently we have a grant from the United States Department of Energy, where we're actually installing a bigger project where we're looking at agrivoltaics in Cranberry, but we're also looking at other projects in vegetables as well. So that's one of the things that I'm working on. The third thing that I'm really interested in is the investigation of canopy management as a cultural practice to improve fruit color. So I've been looking at mowing, pruning, and sanding just to see how they improve light penetration into the canopy, as well as how that improves the fruit quality as well. So I'm going to talk a bit about that at the end of the presentation. One of the main things that I've been looking at in my program has been the evaluation of plant growth regulators. So with my research, I'm trying to use plant growth regulators for a variety of tasks in cranberry production. Because I come from a tree fruit background where they use plant growth regulators for everything from fruit set to quality. That's what I'm trying to replicate the same in cranberry. So I've been looking at improving fruit quality. I've looked at color and firmness. Currently, I've discontinued the work on fruit firmness. I've looked at improving fruit set in cranberry. I've also looked at controlling excess canopy growth. And then I've also looked at improving the canopy establishment after the bog renovation. So those are some of the projects. Most of these are still like early projects, just exploring the use of plant growth regulators. So I'm now going to talk about the improvement in fruit quality, focusing on some of the color research that I've been doing. So like I mentioned before, I started this research in 2019, and my objective was to see if I could improve fruit color using abscisic acid. So what I did was looked at different application rates and timing of abscisic acid to see if that can be used to induce anthocyanin biosynthesis for optimum red color. The idea for the research came from previous work that has shown that the application of ABA at Verizon, which is color break, has been shown to improve red color in grapes. So I thought maybe we could get similar results in cranberry. The picture on your right shows that the differences in color between treated and non-treated grapes. As you can see that abscisic acid actually works to improve fruit color in grapes. So my question was, would it work for us in cranberry? So in 2019, the research that I did was focused on just two cultivars. I did Stevens and House. Then I looked at three concentrations, which and also did two applications, which was a single application at color break or a double application at color break and halfway between color break and harvest as well. So these are the results from 2019 on fruit color in Stevens. This graph shows the total anthocyanin content and then shows the difference between control and treatments. As you can see, there were no significant difference, which was a bit disappointing. So I was expecting this to work in cranberry. And then I also looked at the results in House. I also looked at the results in House, which were very similar to what we observed in Stevens. There was also no significant difference in total anthocyanin content between the treatments and the control. So my question was now asking myself, like, why didn't ABA work in Stevens and House Cranberry? So going back into literature, I looked at some studies that were done in the 1980s and early 90s. It shows that the surface morphology of cranberry cuticle is not very conducive for penetration of sprayed chemicals. So apparently cranberry has a very thick cuticle that doesn't allow rapid diffusion. So based on this, my conclusion was now that the future studies should focus on finding ways of improving penetration into the cuticle. And then I was also going to try other plant growth regulators. That was based on the initial study that I did in 2019. So moving on to 2020, what I did was to increase the number of plant growth regulators that I tried. So I added Ethephon and jasmonic acid. And with Ethephon, I ended up with two different formulations. So I'm just gonna call them Ethephon A and Ethephon B. And this was supposed to have different uptake effects, so they were different in their formulations. The Ethephon was applied at 4 and 8 pints per acre, and then the jasmonic acid at 280 and 480 parts per million, the abscisic acid at 1,000 parts per million. These were applied with the recommended adjuvant, which was just a wetting agent for the Ethephon, as well as a non-surfactant, nonionic surfactant for the jasmonic acid and abscisic acid. These experiments were then repeated with 3 other adjuvants that I'm going to talk about eventually. So last year, because we didn't have a lot of people helping us because of the pandemic, I only did this experiment on Stevens Cranberry. So in terms of expanding the adjuvants, because one of the things that I wanted to try to do was to try and increase the penetration. After some consultation with people in the industry, I came up with 3 additional adjuvants that I wanted to test out with my plant growth regulators. So these are Herbimax. This is a premium oil surfactant that has been shown to increase herbicide activity. I also looked at MSO, and this is a methylated seed oil that has been shown to increase penetration without disrupting the cuticle. And then also at Veda, I looked at Veda, which is a proprietary adjuvant that has also been shown to increase penetration. So the idea between trying these different adjuvants was to try to see if I could get anything that could improve the efficacy of the plant growth regulators that I tried. And then another thing that I did in 2020 was instead of just the traditional TAcy analysis, I also did the DigiEye analysis with ocean spray. So this is an image analysis where you look at fruit color, then it gives you 5 different classes. For those not familiar with the DigiEye output, this is the image that you get from ocean spray. And this is an image, this shows 5 different classes. You've got class 5, which is a deep dark cranberry pixel. And then you have a white or green uncolored pixel. So that's just the image that you get from the DigiEye analysis. Moving on to the results from 2020, these results were actually very encouraging compared to what I saw in 2019. So the application of Ethephon, all the plant growth regulators that I applied except for abscisic acid in conjunction with Herbimax, they were very efficacious in improving total anthocyanin control. And in the best treatment that we have, which is the Ethephon at 8 pints per acre, this was actually like a 71% increase in total anthocyanin content. And you could also see there was a red effect where in terms of the highest concentration of Ethephon at 8 parts per acre was very efficacious compared to the 4 parts per acre. So this was very encouraging to see. And then moving on to the data from the DigiEye. So one thing I should note is that according to ocean spray, there's no correlation between DigiEye and TAcy. What I did with the data from DigiEye, this graph that I have, it shows the percentage of fruit in each color. And if you look at the top, I also looked at the percentage of fruit above class 3 because I assumed anything above class 3 is what is considered desirable for us. So for example, in the control, we only had 94% fruit above class 3. But if you look at one of our best performing treatments, which is the Ethephon 8 pints per acre, you had 98 percent fruit above class 3. So that's a 3% increase in fruit above class 3. And those, all our top 3 treatments were the ones that had Ethephon in them as well. So this is just to give you like an actual image that you get from DigiEye. So on the left is the image from the control, and on the right is an image from the Ethephon at 8 pints per acre. You can actually clearly see the difference in fruit color from the treatment. So you see how it works by getting a better color from that. So looking at the other experiment from this is with the MSO, or the methylated seed oil. With this, in this case, only the Ethephon treatments significantly increased TAcy compared to the control. The abscisic acid and the jasmonic acid was not very efficacious. And you could also see the red effect, where the higher rates at the 8 pints per acre, they had better results compared to the control. So that was really good to see as well. And then in terms of the MSO, if you look at the DigiEye results, in this 88% fruit above class 3. And if you look at our best performing treatment, that was the Ethephon 4 pints per acre, that had 95.8% of the fruit above class 3. So that was really almost a 7.1% increase in fruit above class 3. So that was really encouraging to see. The combination of MSO with the adjuvants that we tried was also very efficacious. I'm not going to show results from VEDA and other adjuvants because of limited time, but based on all, after looking at all the results that I got, so what I found that what works best is the Ethephon B, the formula, because that's a different formulation, and then the Ethephon A, I mean, it's almost similar. Then you also get the abscisic acid and the jasmonic acid as well are working. In terms of the adjuvants that I've tried so far, I think Hebimax is proven to be the best, and then you have MSO and VEDA, and then also LI700. I didn't include it here, but LI700 has also been shown, I mean, it's also been shown to work very well in my experiments as well. So building on that, what have I done now in 2021? I do not have time to show those results, but what I've done is I've reduced the number of treatments from those that proved most efficacious in 2020. And what I've also done is I've now increased the number of varieties that I'm testing. So I have Malika Queen, I have House, and Stevens as well. Unfortunately, this year for us, I don't know, in Wisconsin, we have high fruit rot incidents. So I think that's going to have a big effect on my results. The image on the picture shows one of the samples that I picked. As you can see, there's a quite high incidence of fruit rot, and I think that's going to affect the results that I see this year. Also this year for me, I'm trying to figure out the chemigation. For us in Massachusetts here, we still use a lot of chemigation. So I'm trying to figure out if we can apply these plant growth regulators through chemigation. So what can we do now? Because this research with plant growth regulators is still going to take maybe 2 more years just to figure out the treatments and maybe hopefully get some registrations. Based on some of the work that I did with the canopy management, what I found out is that although not significant, pruning your canopy to just open up the canopy and let the light through actually increases TAcy, or increases the total anthocyanin content. I also did a similar analysis with Gina. So this is things in my collaboration that I'm doing with One, where we're just looking at the image analysis. This was before DigiEye, and we've got similar results where if you do canopy manipulation, you can actually improve the color. In this case, the mowing treatment was not like the very severe mowing that we have. We still left some fruit on the canopy as well. So canopy manipulation could be one of the things that you could do right now if you have a thick overgrown canopy, just to try and improve the fruit color as well. And then this is some data that I got from Thierry at Rutgers in New Jersey. He showed that there's a negative relationship between increasing weed biomass and fruit color. So this graph shows fruit color ranked from 0 to 10, of which 10 is a deep red color. As your weed biomass increases, you get a reduction in your fruit color. So this makes sense because, especially if you have the kind of weeds that shade the canopy, you could actually be shading because you need light for the formation of anthocyanin. So Weed management could be one of those things that you currently do right now, especially if you have those weeds that have the propensity to shed your canopy. So that's one of the things that you'd be able to do now. Like I said, I've done some other work with firmness. I didn't get great results with that, so I don't think there's any need to share with that, but I think this is all that I've been doing so far. And I'm right now I'm expanding this work to look at more ethylene precursors as treatments for plant growth regulators as well. I would like to thank all those people that have helped me in my lab. There's been a lot of people that have helped me, as well as some collaborators from Wisconsin, one in Matthew, and as well as Ed, who has provided some of the varieties that we are testing in Massachusetts as well. So that's really exciting for us, and thanks to him. And then all the funding that I've been getting from the cranberry industry, as well as the collaborations from Ocean Spray and some products from the following chemical companies. Thank you very much.
Thank you. I will take a couple of questions specifically for Giverson, since Sandra and Jeff both had a couple of individual questions, but also now's the time to start asking questions from anything that you've heard during the presentation, including questions of your fellow growers. I appreciate everyone sharing. I guess I can put my screen back on for the final few minutes here. Yeah, we're in peak question hours, so I would love to hear. We had, I guess, a few. All right, so Giverson, are you concerned about the effect of the Ethephon application on fruit ripening and on firmness?
So from the results from the first year, we did not see any effect on firmness, but also because if you look at this application, we're doing it very late in the season, but I didn't see any effect on firmness. I measured firmness with FemTech. I know one thinks we could do much better in terms of how we measure firmness, but I used the FemTech to measure firmness and I didn't see any effects.
We had a lot of discussion on firmness on Wednesday at the grower panel, and I believe Amaya and I are going to be making a video interview shortly to kind of talk about the state of the art, so everyone can stay tuned for that. I know that's a topic of greater and greater interest, both for growers and for processors, and I guess for its effect on processors. Further questions? We've got Giverson here, we've got Jeff, and we've got Sandra. I want to thank everyone for joining us for this lunch hour. Are there any other results that you want to share from the other PGR studies? This is again to Giverson.
I think the results from the fruit set were really interesting. I was able to improve fruit set by using a gibberellic acid, but unfortunately, the return bloom just gets queued, so you don't get any return bloom. So I'd advise not doing it, because I was excited when I saw like you can increase fruit set by up to 50%, which would be great. And also the extra fruit that you said doesn't really size up, so you get a lot of smaller berries as well. So I think that's very interesting in terms of the fruit set. If you could find a way of sizing up the extra berries that you said, that would be really a nice yield increase. But considering the effect that I've seen on return bloom, I would be very wary of using gibberellic acid for increasing fruit set.
I also had kind of an off-topic question. Anyone know of any effective treatment for getting rid of dewberry? I know that is not a specific point of knowledge for any of our speakers, but if anyone knows offhand, that can be answered by a grower too, if anyone wants to type in something they've been doing. But Amaya did respond to Giverson's answer that we've seen the same here, as we've done similar research in Wisconsin on the PGR studies. I'm happy to take more questions. We are coming up on the end of the hour, so I'm going to make one more reminder. If you haven't spoken with your tax preparers yet for the new tax changes coming for the coming year, please do that so that you are ahead of the game. So thank you, Tom and Alex. Obviously, the University of Wisconsin Division of Extension, but then a special thanks to Sandra from Fraser Valley University, to Jeff from the USDA in New Jersey, and then from Giverson from UMass. Thank you guys all for connecting with us today and keeping us abreast of everything new in pollination, in our pre-breeding genetics program, and in fruit color and quality that's going on throughout the world. Great thanks to everyone.